Sí, ¿verdad? Ana. Buenas tardes. Buenas tardes. Gracias por acompañarnos en esta conferencia magistral, la del mes de abril, en este mes que hemos dedicado durante los festejos de nuestro 25º aniversario a la División de Ciencias Ambientales. Hoy tenemos como invitada a la doctora Anca Delgado, quien es profesora de la Universidad Estatal de Arizona, a quien le agradecemos que haya atendido la invitación de la división y obviamente del instituto para dictar la conferencia Marte Rojo, Marte Verde, el papel de los microbiomas en la detoxificación, descontaminación del biointemperismo del regolito marciano. Así, eso sería, ¿no? La conferencia encaja perfectamente en la visión que las organizadoras del simposio, el 15º simposio de ciencias ambientales diseñaron para este evento. Nuestra vida entre microorganismos. A lo mejor tuvimos alguna vida anterior en Marte y quién sabe qué haríamos por allá. A lo mejor todavía hay por ahí algunos restos de vida microbiana, pero bueno, eso nos lo platicará Anca Delgado, que como les digo, viene de la Universidad Estatal de Arizona. Una breve semblanza de su biografía académica es la siguiente. Anca Delgado es profesora asociada en la Escuela de Ingeniería Ambiental, Ingeniería Sustentable en la Universidad Estatal de Arizona, y su laboratorio se enfoca en procesos microbiológicos que remueven contaminantes o que proporcionan productos de alto valor químico para la sociedad. Su investigación emplea microbiomas naturales y sintéticos para transformar, degradar o secuestrar contaminantes químicos, para mejorar la calidad del suelo y del agua, o para producir productos biológicos a partir de corrientes de desecho. Entonces, como pueden ver, el tema que nos trae nuestra conferencista del día de hoy es un tema por demás importante. Hoy nos hablará de Marte, pero muy probablemente todo esto se aplica también a nuestro planeta y a lo que hemos estado haciendo con él y a lo que deberíamos de dejar de hacer, ¿no? Así es que bienvenida, Anca, muchas gracias y pues todo tuyo el auditorio.
Buenas tardes, voy a empezar un poquito en español. No hablo mucho, pero entiendo mucho. No voy a poder dar esta presentación en español, pero quiero que estoy bien agradecida de la invitación. Muchas gracias a profesora Ontiveros y a IPCIT por esta invitación. Y now we're going to switch to English. Pero al final, si estás más cómodo de preguntar las preguntas en español, puedo tratar, y si no entiendo, profesora Ontiveros me puede ayudar. Okay. So again, welcome everybody. It's a pleasure to be here. Thanks everybody here that's in person, and thank you very much to those who joined online. I will talk to you about a kind of peculiar project. You're going to see that we are trying to tackle problems maybe of the future, or problems that do not yet exist, and maybe they're not even of this Earth yet, but certainly, as was explained, they are applicable to Earth. And so I'll begin just with the acknowledgement and kind of to set the stage here. This project is really a big effort of 3 universities: Arizona State University, the University of Arizona, and Florida Institute of Technology. We are very grateful to the National Science Foundation and to NASA, who invested a lot of funds for us to tackle this challenge I'm going to talk to you about. And most of the work that I'll show you today was done by Alba Medina, my PhD student, and Brianna Pais, also with collaborations and work by Andrew Palmer's work from Florida Tech and Malak Tefeli from the University of Arizona. All right, so here we go. So it is predicted that human trip to Mars is likely to happen in the not so distant future. So NASA has been thinking that we might be getting on Mars by the 2030s. Other entities like SpaceX have even more ambitious sort of trajectories. We might be there even earlier than that. And so we are preparing for that human mission to Mars. And the question is, of course, why do we want to go to Mars? There are obvious sort of scientific questions. We believe that Mars may have been a crater for life, or then life might have existed, and those signatures are still there and we can study them. There's the question of testing technologies that developed here on Earth on other planetary bodies. But then there's also the question of, well, perhaps there could be some mining of resources that could happen on Mars, and even thinking way ahead of perhaps Mars could be a way to resolve some of the overpopulation challenges on Earth. But regardless, we have been thinking for going to Mars for many years, and we most likely are going to get there one day. And so we have learned a lot about Mars, especially from the missions that touched down on Mars, that have sent rovers all the way from the 1970s, the Viking missions. And this is an image of, or a map of the Jezero crater. This is where most of the rovers have touched down on Mars. And so the Jezero crater is a crater that is thought to have been filled with life, with water, and has sort of delta-like rich clays. And the reason we have the Perseverance rover active there is to look into the possibility of signatures for life on Mars. You may have seen the Mars 2020 Perseverance rover images. I've sent very beautiful pictures of Mars. So it searches for signs of ancient microbial life to advance the quest to explore the past habitability of Mars. It is also a rover that is collecting Mars samples, collecting rocks, collecting regolith, which is the term for the surface material, basically a combination of broken rock and soil. And so those samples are being collected, and the vision is that there's going to be another mission in which another rover will be sent to retrieve those samples and bring them back to Earth. And that is projected to also happen sometimes in the 2030s. So I wonder who'll get there first, the samples from Mars to Earth or humans to Mars. So that remains to be seen. And so here's where the Perseverance, the map of where it has traveled, I think somewhere on the order a bit more than 33 kilometers. There's also on this map an image of the helicopter on Mars. This helicopter is the first one to fly on another planetary body. So it's shown that even with the less dense atmosphere of Mars, you can still fly a helicopter. And so along the path, the rover has collected a bunch of regolith and other rock samples. And here are some images. The rover has already collected 28 out of the 38 projected samples. Sent some beautiful pictures of very interesting formations that it has discovered. And so we look forward to getting those samples eventually on Earth. And so while we think about what the information that we learn from the rovers, the vision is that if a human crew goes to Mars, then we have to be able to get food and supply those people on Mars while they're there. The trip might take on the order of 6 to 8 months. Obviously, we cannot rely on shipments. That could cost life. Also, if you think about traveling or such distance in a ship, space is at a premium. And so then the question is that if there are people there on a scientific mission, how expensive it really to supply them from Earth. And so, there was some estimation back in 2015. People said that if we even want to send a letter, it might cost on the order of $17,000. But what if we want to send some food? Let's see, if we have 5 ounces of arugula, it would cost $50,000. So that's the most expensive salad. But then 50 ounces of arugula, 5 ounces will only have 50 calories. So something like 2,000 calories with arugula would be worth $500,000. And we don't even know how many calories would be needed for humans on Mars. But the idea is that fresh food cannot be supplied. And a human mission on Mars has to rely on being able to grow their own food. And so maybe hydroponics could be an option, but many plants do not do well hydroponically. Think about sort of root plants. And so the question is, can we grow plants in the Mars regolith? And so Matt Damon did it in The Martian. And so surely we can think about if we do it. It would be imperative to be able to grow plants in a regolith sort of type of agriculture. Water is going to be obviously scarce on Mars, and it's going to be very costly. And so the idea is that you have to somewhat prepare to have both hydroponic and regolith-based sort of food production. So is it as simple as Matt Damon showed it in The Martian? Can we just put potatoes in the regolith and start growing our plants? Well, it turns out it is not. And a major challenge with the Martian regolith is that it contains the chemical perchlorate. So back in 2008, when the Phoenix lander touched down on Mars, it had a very nice wet chemistry lab on one of its robotic arms. And here shown in this image is the robotic arm. And here are some regolith samples that were mixed with water. And what those analyses showed is that a high concentration of perchlorate, which was then converted to a concentration of mass perchlorate per mass of soil. So Mars regolith contains on the order of 0.5 to 2.8%, so between 5 and 20 grams of perchlorate in the regolith. Perchlorate has been detected at calcium, magnesium, and sodium salts. And as you may know from Earth, perchlorate is a regulated contaminant. In drinking water, it has low maximum contaminant levels. And in Arizona, it is 7 micrograms per liter. And so it is toxic, it is regulated. And this is a major challenge that was not expected. Perchlorate concentration is also orders of magnitude on Mars than it is on any Earth soil. And so this is really something that has really put a spin on the challenge of can you grow plants in regolith and how that material could be used. And so where's perchlorate coming from on Mars? Well, there are believed 2 major hypotheses. One is believed that gas phase oxidation of chlorine in the atmosphere with either ozone or oxygen formed perchloric acid, which just deposited on the surface. And then a second hypothesis is that there were mineral UV oxidation of chlorine in the surface material, which led to the formation of this perchlorate. And then there's a suite of all sorts of redox reactions. So the environment is very oxidative, so a bunch of oxidation reactions and a complex chemistry that involves both perchlorate and sulfate, which is also present at very high concentration on the order of 40 grams per kilogram of regolith on Mars. And so the challenge then becomes, what can we do to remove this perchlorate from the regolith? This is something that has been sort of a black box. NASA maybe has planned to send humans to Mars, but there hasn't been a good approach or technology that they will use when we get there. And so in the meantime, while we wait for those samples from Perseverance to arrive to Earth, we, on any sort of experiments, plant-related, microbial-related, have to rely on what they're called simulants. So Martian regolith simulants are typically man-made materials from various sources on Earth that are meant to reflect the physiology and the chemistry and the properties of the Martian regolith that have been sent back to us from information from rovers. And so right now, there is one regolith called Mars global simulant that is produced even at the commercial scale, that is considered the most mineralogical accurate regolith. It has a mixture of crystalline and amorphous materials. So it's meant to simulate both the mineralogy and the bulk chemistry of what we know from rover information that regolith should look like. And so what do we know so far about growing plants in Martian regolith simulants? So this is work that was done by my collaborator, co-PI, Andrew Palmer at Florida Tech. And so what his group did, they took three Martian regoliths. They took one that was developed by NASA's Johnson Space Center called JSC, another one called MMS Mars Mojave Simulant. This is one that's directly sourced from the Mojave Desert. And then took this Mars Global Simulant, the one I just showed you that is considered the most mineralogically accurate, and looked at plant germination experiments using lettuce and tail cress. And so what they saw is they also added perchlorate. So the conditions with P showed that perchlorate was added. And so they put these seeds into two of the materials, to JSC and MMS, saw germination with or without perchlorate in the regolith. But then notice that when the Mars Global Simulant that is considered the most mineralogically accurate, there was no germination, zero germination for both of these plants with or without the perchlorate. So then the question is, why is it that finally, when we created a simulant which we believe really reflects the chemistry on Mars, why can we grow the plants? Well, we cannot grow these plants in part because there is no organic carbon in these materials. There's very poor nutrients. And there's toxicity from the perchlorate. But then here are some data. What happens if you do not provide nutrients? This is with the JSC and the Mars Mojave simulant. So the viability of those seeds, it dies off in just a matter of a little bit more than a week. If you do supplement with nutrients, you can get them to be viable. And here are some images of the simulants with, this is tail cress and lettuce, some plants that were growing in the simulant. But the idea is that in no matter what simulant was used, if perchlorate was added, all the seedlings perish within 5 to 9 days. So no full plant was able to be developed. But this is telling us that it's really challenging to grow plants in these materials, and there is probably a combined toxicity from perchlorate, salinity, and the physical properties of the regolith. But before we look into some more of those, we have to think about, perchlorate. The issue of perchlorate has to be addressed. This is something, it's a chemical that is toxic to humans. If it's uptaken in the plant biomass and humans would consume it, that would be problematic. Also, inhalation of perchlorate particles would be dangerous. And so maybe, the technologies for perchlorate are somewhat limited because perchlorate is somewhat difficult to do any sort of oxidation or reduction of it. It's highly soluble, so you could consider, washing it, but who is going to go to Mars to wash dirt? So then you could grow plants, right? So you can grow plants. And so one of the most sort of efficient technology, biological technology, biotechnology that exists is microbial perchlorate reduction. So somehow microorganisms have evolved this very nice pathway to reduce perchlorate. Perchlorate can be used as their electron acceptor. In their metabolism, acetate is a common electron donor, and so perchlorate is reduced to chlorate and chloride, and then eventually to chloride and oxygen. And so some of the enzymes involved are perchlorated chloride reductase and the chloride dismutase. And then in the end, this oxygen is actually being reduced to water. Most of the perchlorate reducing microorganisms would love to be aerobes and would consume oxygen if they have the opportunity, but in the absence of oxygen, they'll take perchlorate as well. And so Bacteria that can reduce perchlorate have been identified in many phyla, and so here are some examples of proteobacteria, including the Chloromonas and Azospira, which are considered somewhat model organism for perchlorate reduction. And so if we are to use microorganisms as the first step to detoxify the Martian regolith, then the question is, can they do it? Can they work with the concentrations of perchlorate that is in Mars regolith? So if you remember, concentration of 0.5 to 2% in the regolith, think about adding some water, let's say 10 to 20%, then the concentration of perchlorate in that water would be on the order of 50 to 100 grams per liter. And so on Earth, we are not concerned with those high concentrations. We are in groundwater for bioremediation application. We're mainly looking at micrograms to milligrams per liter. But there have been a few efforts to look at higher concentration of perchlorate and see whether that can be microbial reduced. So this is some work that was done at the University of Nevada. And so in these sort of experiments, they track very nicely the kinetics of perchlorate reduction, and they looked at concentration up to 2.5 grams per liter, so 2,500 milligrams per liter. And so the inocula that was used here was return activated sludge from the secondary treatment in the wastewater treatment plant, but also anaerobic digester sludge. So what it was, we know that perchlorate reduction is possible in a matter of just a couple of days. But notice that if you do have some high salinity in the system, then your kinetic rates really become much smaller. It takes the time on. Now we're talking about days to weeks, and then at 2.5 grams per liter, we're even seeing a stall with the return activated sludge. And so concentrations matter. So to do this kind of effort, you also need to have a microbiome or microbial community that is able to tolerate very high concentrations. And so with all that prelude of kind of the putting the pieces together, a couple of years ago, this is a new project in my lab, but a couple of years ago, we put together a team from the 3 universities. And we said, okay, this is how we envision that we might be able to tackle the problem of perchlorate in Mars regolith. So we have the Mars regolith, which has concentrations of perchlorate. The first step we would do is sort of a detoxification, so conversion of that perchlorate to chloride in oxygen and water. We know that in that process, we would grow some microorganisms. And then these microorganisms could be considered a source of primal soil organic matter. It's microbial biomass and residues. So that would provide, some organic carbon in the system and maybe increase the fertility of the soil. But then obviously that would need to be later, stabilized and diversify. We would also need to understand what is sort of the makeup of the soil organic matter, because we are envisioning now formation of soil organic matter that are in conditions that are not typical of how it would occur on Earth. And then we would have to be able to check that indeed you are able to grow plants, that you've removed toxicity, and have the right potential to use the regolith material for plant growth. So I'll start with telling you more about the perchlorate reduction part, which is the work that we do in my lab. And so the first simple exercise we did is sort of an enrichment exercise where we said, okay, if we want to have perchlorate reduction and we're going to be working with concentrations on the order of grams per liter, we have to have also some good inocula. And so we took sodium perchlorate, we made some sort of microcosm experiments. We used inocula, we used some desert soils that were collected from Arizona, including one from the city of Goodyear, from a Superfund site that is contaminated with perchlorate. And then we also used, knowing from the literature that the activated sludge in the wastewater treatment has some pretty good perchlorate reducing microorganisms. We also tested that. We used sort of a nitrogen headspace and anaerobic conditions, and then we provided acetate, glucose, that's ethanol, as the electron donors. So what we saw in some of these first experiments, what you're looking at, concentration of perchlorate on the y-axis and time on the x-axis, and then you're looking at the various inocula we tested, and starting with that mixed liquor, wastewater sludge, tropical soil, desert soil, and with ethanol, acetate, and glucose. But what we saw in just a matter of days, we saw perchlorate reduction activity with the wastewater sludge. By day 28, perchlorate was no longer detected. The soils took significantly longer. We only detected perchlorate reduction when we measure at day 100. But the idea is that, you know, perchlorate reduction at these very high concentrations, higher than you have seen in the literature so far, is possible. Depending on your inoculum, you might see some sort of lag phase, but eventually most of these inocula were able to, with the selective pressure of the high concentration of perchlorate, were able to reduce it. And so we really like this mixed culture that was developed from the waste activated sludge. If we think about, you know, a mission on Mars and what kind of microbiomes and microorganisms we'll take with us, surely we'll take our microbiomes from the gut, which contain some perchlorate reducers. We're also going to generate waste, which is a place where these sort of microorganisms enrich. So we decided to really move forward in some of our experiments with this mixed culture. And so we did some initial characterization of what, you know, from the initial that was sampled at the wastewater treatment plant to the final that was enriched after incubation with 4 grams per liter perchlorate. We saw enrichment of Rhodocyclaceae and Pseudomonas type of microorganisms, which are known to do perchlorate reduction. You may remember that perchlorate on Mars is found either as a sodium, calcium, magnesium salt. So we also grew the culture with these 3 types of perchlorate salts, or a mixture where we added 1/3 of each sodium, calcium, magnesium. And so we saw that there's no issues with growing it with either form of perchlorate, although we did see significant precipitation in the system when we used the magnesium and the calcium form of salt. Okay, so now we know we have a good culture, and now we can really, you know, try to see what we can do with the Mars regolith. And so we made a special Mars regolith in our lab. We purchased the Mars global simulant, but we added perchlorate. So no Martian regolith simulant is available to be purchased with perchlorate in it because of issues related to human safety, and because you have to have a special laboratory to be handling like that. And so we made our in-house perchlorate containing regolith. Concentration was 1 weight %, so 10 grams per kilogram. And then we set up a series of experiments. We used our mixed culture that we developed from the wastewater sludge, but we also had some pure cultures. Halopherax denitrificans is an archaeon, not a bacteria. This one can grow at very high salt concentration, even higher than seawater. And then we also had a strain of Dichloromona denitrificans, one of the common perchlorate reducers. We used anaerobic conditions typical of the Martian atmosphere. So of the Martian atmosphere is about 95 CO2%, 3% nitrogen, 2% argon. That's the major composition. And then we use acetate as the donor. And so what we did in this case, so our regolith material was at 1 weight % perchlorate. And so for us to control the perchlorate concentration in the water, we had to play with the amount of water in the system. And that is, so we wanted to look at 1, 2.5, 5, and 8 grams per liter of perchlorate in the regolith water. So we added different amounts of water to these bottles, whereas the one that had the most water was at 1 gram per liter. The one that has the least amount of water was at 8 grams per liter. But the one that also had the least amount of water had also the highest concentration of everything else, including the salts in the regolith. And so what we saw in these experiments, again, a concentration of perchlorate as a function of time, and 1 gram per liter, we're using the mixed culture, we're seeing very nice reduction. The kinetics are slower than what we observed in regolith-free cultures, but we are able to completely reduce that concentration. 2.5 grams per liter, also similar results. We had a bit more variation in this experiment, but we basically got rid of about 95% of the concentrations. 5 grams per liter, this is where we saw actually an artifact where high concentration of perchlorate also gave some sort of competitive advantage to the mixed culture in this case, and the reaction, the kinetics were quite fast. But then at 8 grams per liter, our mixed culture barely showed any activity, maybe about a couple % reduction. Now notice we also use this Halopharax strain shown in green, which I told you it's an archaeon that can tolerate high concentration of salt. So this one did also fairly well at 5 grams per liter, and it's the only one that had some sort of significant reduction of perchlorate at 8 grams per liter. Now our Dichloromonas culture that is operating under normal salinity conditions, we did not see a difference between that and the mixed culture. So it confirmed to us that perchlorate reduction at 8 grams per liter, at least in this case, is inhibited most likely by salinity. Then when we looked at some of the reduction rates that we achieved in the system, so highest reduction rate, about 315 milligrams per liter a day were obtained under 5 grams per liter perchlorate. So to our knowledge, this is the highest perchlorate reduction rate reported in the literature so far, because we're using concentrations that are also very high. And then we also now have information that some microorganisms that can tolerate high concentration of salt might be better adapted to dealing with under these conditions because of the high salinity of the regolith itself and the high salinity that comes with having high perchlorate concentration. And now, of course, we are interested, especially if we're using a mixed microbial community, and what is the composition of that microbiome that developed. And so if we think about practically, if we are going to do some sort of detoxification on Mars, you can think of maybe some of the microbial community composition or the microbiome being somewhat of a primal microbiome that is expected to develop under those very selected conditions. So anaerobic conditions, high concentration of perchlorate, high concentration of sulfate. So then we took those regolith conditions and we analyzed the microbial community. We had the various concentration of perchlorate in the experiments, 1 to 8 grams per liter, and then we look at 2 time points in the experiment, time 0, day 21, and day 60 when we closed the experiment. So what we saw is, the microbial community really represented the major processes, metabolic processes occurring. So this bright pink here is a strain of Pseudomonas balearica that really had enriched, and it was under higher abundance under conditions where perchlorate reduction was the most extensive at 2.5 and 5 grams per liter. We also saw a bunch of sulfate reducers. So more than 50% of our community was sulfate reducers. This is consistent with the fact that the Martian regolith contains, as I mentioned, 4 weight percent sulfate, which is added as magnesium sulfate in this case in the Regolith simulant. We also looked a little bit at sulfate reduction, and we saw evidence of sulfate reduction in this condition, but notice that at 8 grams per liter, in the conditions where we had 8 grams per liter perchlorate, we also had 16 grams per liter sulfate. And so, the microbial community is mainly representative of that sulfate reduction. But anyway, we got a clue of, what this sort of initial primal microbiome might look like. Okay, so we know now that perhaps the perchlorate issue can be resolved with microorganisms. We tested concentrations that are some of the highest that we probably haven't had time to think about here on Earth, but we could. Especially if we're talking about, brines from perchlorate treatment systems. So now we have some evidence that perchlorate reduction can be done at very high concentrations. And so what happens next now that we have reduced the perchlorate and detoxified the regolith? Did we somehow improve the properties of that regolith? Did we make some sort of soil organic matter? And so the first, sort of assessment we looked at is just to measure the total organic carbon concentration in the system. So what you're looking at is percent carbon on the x-axis and various samples in which we looked at on the y-axis. Here is the Martian regolith simulant with perchlorate as we started, and a biotic control, and then the amount of organic carbon that was formed in this, or that was measured in these regolith experiments after perchlorate reduction activity. And so as you see, a very nice increase in total organic carbon in the system. And then we also compared them to the 2 desert soils that we had initially used for some enrichment cultures. And so we're not quite there to, just by doing perchlorate reduction and maybe some sulfate reduction in the system, we're not quite there as a typical soil from a desert, which I think it's a good comparison, but I think we're getting there a bit closer. But then the second question is, what is in that soil organic matter that we form? And so for that, this is now work that was done at University of Arizona in Malek Tefayli's lab, who's a world expert on soil organic matter characterization. So we did some bulk organic matter characterization using this technique called FTICR-MS. And so what you're looking at here in just this first is just an overall metabolic profile grouping, an MDS ordination that looking at the samples in which we use that mixed culture from the wastewater sludge, the Halophorax archaeon culture, and an abiotic control. And then you see a very nice grouping as a function of inoculum. For the wastewater culture, we also see a pattern related to perchlorate concentration, in which one is somewhat distinct, this one being 1 gram per liter, 2.5, 5, and 8 grams per liter. So these are some initial profiles we observe. And then the question is, what is the sort of class and what are the major categories of this primal soil organic matter that we are formed? So again, we looked at the archaeon samples with the regolith and the ones with the mixed culture from the wastewater sludge. And so some trends that really popped up here is that the class composition, so you have amino acid, sugar, carbohydrates, condensed in unsaturated hydrocarbons, you have proteins, etc. Those would be typical. What we see here is that all of the samples are predominated by lipids, so lipids being the major class. Now, this is not how, if anyone is familiar with soil organic matter, that's not how your typical distribution in a normal earth soil would look like. Some other trends that we saw were related to perchlorate concentration. We noticed that the relative abundance of phenolics, which is labeled here as lignin. Lignin is, there's no plant material here, but sometimes that class is called lignin. So phenolics in this case increased as a function of perchlorate concentration. So these type of phenolics could act as antioxidants, and we believe because of, as you increase the perchlorate concentration, you also increase that oxidative stress, and so this could be an artifact of that very unique environment. We also saw that proteins, shown here in red, were also their relative abundance decreased as a function of perchlorate. So as an example, here would be at the concentration at 1 gram per liter perchlorate with a mixed culture versus 8 gram per liter. And so this maybe is an artifact of denaturing of proteins, because also, again, the high perchlorate concentration and the high oxidative environment. But at least, we get a sense that what could be the starting material, and of course, this sort of primal organic matter then could be stabilized and diversified. Remember, we are doing experiments, or the formation of these organic matter is under anaerobic conditions, without oxygen input and without addition of any sort of plant material. And then one could even, look at various ways in which you can try to extract more information out of this kind of metabolomic information. So we looked at some of the metabolites feature. This is for the mixed culture. What is common and what is different between the various conditions. So here I have in red 1 gram per liter perchlorate, 2.5 in green, 5 in yellow, and 8 gram per liter perchlorate in brown. So we saw some 7, 11, and 43 metabolically unique features at various perchlorate concentrations that we tested. But then we also saw some common ones. So, some of this information is telling us that there may be some kind of specialized profiles that are associated with higher concentration of perchlorate. But then there's also some commonalities between these features. So we are still learning more about this, but the metabolomics that related in the soil organic matter composition. But at least we know that what we're working with now. And so now the final sort of push is, well, we've gotten rid of perchlorate. We have formed some primal organic matter. Can we grow plants? Did we infer fertility to this regolith? And so that is work that is being done by Andrew Palmer's group at Florida Tech. So at Arizona State University, we prepared the regolith. We did all the bioweathering, detoxification experiments. And then we shipped these samples to Florida Tech at Andrew's lab. Andrew Palmer is an expert in growing plants in lunar and Martian regolith. And so we provided various samples, the ones that we grew, the ones that from the experiments with mixed culture from the wastewater, and then Haloferax denitrificans, the Archeon. And so they were going to basically do some initial characterization of these regoliths, to look at some scanning electron microscopy, and some characterization data, and then screening for different germination species, and then isolate the surviving plants and characterize them. So it would be important to look at those. So first, let's look at a little bit what has changed on the physical properties of the regolith. So this is the top two images. You're looking at the regolith as it was before it was used in sort of any bioweathering experiment. The soil particles of this Martian regolith simulate are somewhere on the order from a couple hundred nanometers to up to 1 millimeter. So it's a fairly fine process. But then, what we're seeing in this regolith is these like lines and edges from the particular rocks that are present in there. Now here are images. I'm not sure how good the resolution is on mixed culture, the regolith that was bioweathered with the mixed culture versus the Haloferax denitrificans. And so what is obvious in this image is this phenomena of pitting, where you're seeing pits or holes now into the regolith, as if it's like sort of a corrosion-like process, which is very common, or it's expected to look for bioweathering. So the physical microbial activity did change the physical property of the regolith. And now we go to the really important question: growing plants. And so soy, tomato, tailcress, lettuce, green pepper, foxmeal millet were all tested for germination. And guess what? Most of them, except the foxtail millet, zero germination. After all this effort, and still we couldn't get that to germinate. But we did get germination of 40%, which is now a sort of a world record. 40% germination in the regolith that was bioweathered with the mixed culture, where perchlorate reduction occurred. And so this foxtail millet, if for the plant experts in the room, you may know it's sort of a drought-resistant, higher salinity-resistant type of plant. It makes grains. It's grown in Asia and other places of the world. And so this told us that perhaps the selection of plants, perhaps one could kind of think about sort of a multi-generational way to look at germination, where you would start with using some plants that are more drought and high salinity resistant. And then you could, second generation, third generation soil, you can try to grow these other plants. But I'll show you, some other images. These are germination. These are the seeds of the foxtail millet. Up here on the top, these are Arabidopsis thaliana seeds that never came through 1 week after planting. And then this is the most we have the most robust plant so far, 5 weeks of the foxtail millet that we've been able to grow so far. So you may think, well, you guys, maybe you suck at growing plants. It's not true. This really is from in a laboratory where they grow all sorts of plants and have many plant projects and a lot of experts. It just has been very challenging to be able to grow plants. And this is not just the group here for our project. This is similar challenges, and perhaps even more difficult are with lunar regolith, where people are having a very difficult time growing plants in the lunar regolith. And there's a major push by NASA to the latest call for proposals from NASA has been to how to improve growth of plants in the lunar regolith. And then similarly, these are some of the best results so far on improving plant growth in these materials. All right, so I'll leave you with some sort of a key messages and food for thought. So we showed that microbial perchlorate is achievable. Like it was really important to be able to say that, okay, at least that aspect of the challenge of using the Martian regolith material, either for plant growth or other application in which humans might be in contact with, I think we have a good grasp on that and we can handle it. You saw the choice of culture, whether, it might depend on the environmental conditions, particularly controlling, salinity, pH, sulfate, etc. Plant growth on Martian regolith still remains a challenge. Now, I do not, for the purpose of today, I couldn't show those data yet, but we have found that we can improve germination from that 40% to significantly bump it up, maybe closer to 80%, if we provide spacer in the regolith. So spacers, think about sort of like perlite or glass beads, inert material that allows, for better root development, for kind of a formation of a different microenvironment. So this tells us there is also a toxicity related to the physical properties of this material, not just the chemical composition. And then, while I've told you that perchlorate is a problem, just know that perchlorate is on Mars is really a resource. So it potentially is the most important resource of oxygen. So if we can somehow harness microorganisms to take perchlorate and reduce it to chloride and capture that oxygen, I think we have some calculations that a kilogram of regolith could produce up to 7 liters of oxygen. And so this is indeed, we have to work with the resources that are available in there. So it is a challenge to grow plants. It is toxic to humans, but is also one of the most important resources on Mars. Okay, so with that, I thank you very much for your attention, and I will take questions. Muchas gracias. Okay, so I'll start with the first question. Thank you very much. Both the water is a problem. So our understanding, and I think the scientific community is in agreement that there is frozen water on Mars. Most likely that water will have also a very high concentration of perchlorate in it. And so if somehow some of that water could be channeled in a way that it could be used, that would be one option. Obviously, we will bring water on Mars. There will be water on the ship. There will be hydroponic systems. So perhaps, some of that water could be channeled to a kind of a small dedicated area where you could provide some water. Now, everything that I've told you today related, obviously, to microbial activity or plant growth, it is on the presumption that the same principles of life would have to apply on Mars, right? We would need the right temperature, pressure. We would need to shield microorganisms, plants from what is it called? The rays, the radiation, etc. And so we envision that we would be able to, or this could be done in sort of either in caves or in encased sort of area similar to the way you would plant a greenhouse, that sort of thing. So some water has to be channeled to this process initially if you want to use the regolith material for plant growth. Water will be scarce, yes, but hopefully maybe some of the water that is on Mars could be used for this process. Then the second question, have we considered the other wild relatives? So the plants that were selected were plants that specifically NASA has requested us to test for two major reasons. There is a large body of knowledge, especially also trying to grow plants on the International Space Station. And so it's a mixture of plants that make fruit, like tomatoes, etc. And so our initial efforts were to kind of cater some of these plant experiments to NASA's needs and vision for the important plants to study. But yes, we are now considering maybe taking a different approach where maybe the first generation plants would be somewhat highly drought resistant, saline resistant. And maybe I'll suggest to my collaborator, Andrew, if we could look for wild varieties of tomatoes that would grow rather than your typical common tomato plant that would be used here. So thanks for that. Thanks. Yeah. Yeah. Mm-hmm. Yep. Yes. Yeah. Yeah. Oh yes, the nitrogen challenge. I think you hit the nail in the head. It's considered a major limitation. So the major source of nitrogen is, from the atmosphere of Mars. As you saw, it's in low concentrations. So the agreement in the sort of space plant scientific committee is that an initial sort of nitrogen as a fertilizer has to be somewhat available and be brought there. We cannot, one cannot think that you could start growing plants without just by using what is available on Mars. So this is really also another challenge of, where can we get more nitrogen from, or what sort of processes are available to better capture and channel that. But yes, this is the initial thought. We have to bring some fertilizer, and then we can, also rely on some of the nitrogen there, but it's limiting. Mm-hmm. Yep. Yes, so Andrew Palmer is looking at some of these. So if you mix the Regolith with some potting soil, that issue is no longer an issue. Spacers is one option. What? Hydroponic gradients. Okay, I think that's not something that we're working on, but that could certainly be an option. Although to the best of our ability, we're trying to stay away from looking at conditions in which water is really plentiful, and so that's we are trying to more just look at Regolith, ideally unsaturated conditions, although We use that for plants, but in our microbial experiments, we did have saturated conditions. And then the other thing that Andrew Palmer's group is looking at is sort of providing now a sort of plant microbiome bioaugmentation with the sort of plant supporting microorganisms to see if that also helps improve germination and then plant development. Yeah.
Hola. As I understood, this you call primal organic matter. It comes from some microorganisms that are using acetate. So in Mars, where this acetate will comes from?
Very good question. So acetate was sort of an obvious choice because wherever humans go, we will have to do some sort of treatment of organic waste, including our own waste. And so the vision is that the source of acetate and other volatile fatty acid would be digestion of waste and organic matter material that would be associated with the crew activities. And so that's another, in the next phase of our project, we will look at some directly providing, treated sort of sludge or some organic material that is a bit more complex. But acetate is one of the most common volatile fatty acids from anaerobic digestion. So it made sense.
Hi, first of all, thanks for the talk. It was amazing. Have you ever tried to clone these perchlorate dismutagens into other kind of bacteria?
So the short answer is no, we have not tried, but we are very much thinking about that. And so one of my PhD student wrote a very nice proposal recently for her qualifying exam of how we would do that. So we are thinking about it. And we are also thinking about, can we engineer some of these microorganisms so that they don't take the oxygen and convert it and reduce it to water? So we haven't done it, but we're thinking about it. And I think with the right expert team and the right thought process, this is something that definitely could be of interest to NASA and to future missions, is going beyond, how we can now extract oxygen out of that perchlorate. Thank you. Very good question. Black yeast, as in molds, black molds. Black. Okay. Yeast. Okay.
They have a melanin pigment, and also they grow.
Yeah, well, thank you for that tip. It's something we'll definitely look into. No, we have not considered, but thank you for that, black yeast. Okay, and they do grow on, they would grow on sugars, or yeah, okay, very nice. Thanks for that. And I know you know a lot about perchlorate reduction and sulfate reduction.
So, I guess my question, so you can expand on the sulfate reducers being present in the microbial community. What do you think about the challenges ahead? What does that mean?
Sure.
When you are trying to bioweather?
Okay, so this is a topic that one of my PhD student, Brianna, is actually almost working on 100% now. So we're looking more on the sulfate reduction angle and competition between sulfate reducers and perchlorate reducers. So I think the major challenges with sulfate reduction is formation of sulfide and then precipitation of metals, or some metals that might be essential for plant or microbial growth then might be sequestered and no longer be available for microbial activity. sulfide is toxic. Sulfide gas, free sulfide is toxic to many organisms. In general, you know, the material, the regolith, is very high in iron, so we are not seeing a lot of free sulfide accumulation. We are seeing a lot of precipitates. I think I may have a picture of showing you we started with let me see if I can go, I have in the extra slides, what happens to the regolith. What starts red and beautiful, and then you put, oh, there it is. Okay, so we started with Martian regolith simulant that looks like that beautiful red color of Mars. And then I don't know if resolution is good enough. You get sulfate reduction and it turns completely black. That's due to the formation of sulfide minerals in there. And so how this translates to, you know, at least downstream for, you know, for plant growth, the group of Andrew Palmer is now looking at, you know, taking the Martian regolith simulant and pulling it apart component by component, removing sulfate to see, you know, how that affects plant growth, etc. Right now, we're just trying to understand, you know, this interplay between the competition of perchlorate reducer and sulfate reducers. In general, I think we're seeing that both processes can co-occur unless you go to very high concentrations of salt, in which we saw mainly sulfate reduction rather than perchlorate reduction. Thanks.
I have two questions for you. It was a good talk.
Thank you.
In your personal opinion, regardless what NASA thinks or needs, when we'll be able to set a colony on Mars? And the second one is, did you consider the gravity for plant growth? Because all the plants grow here.
Yes. So a colony on Mars by some experts, including one of my collaborator, Andrew Palmer, believes that it might happen in the next 50 years. I'm somewhat in agreement, maybe. And so I think the fact that we are here today discussing something that, you know, an environmental process that we've known so much in bioremediation, and we haven't really thought to explore this for Mars, is a bit unbelievable thing, you know, with the vision that we might go to Mars soon. But regardless, you know, I think 50 years is what is projected. In my opinion, I don't think it might happen. It will happen yet in 50 years. And then your second question was, have we considered gravity? Yes, we have considered gravity. So at Florida Tech, we have a special built chambers that can put microgravity conditions. So we are looking at both microbial and plant experiments under microgravity. Now, you may be aware that, you know, plants have been grown in the International Space Station, and so in environments with lower gravity than here on Earth. And so we do have some sort of kind of information about what are the possible consequences. In general, we're seeing, you know, smaller plants, you know, lower yield under microgravity conditions. And with some of the microorganisms that have been sent to space and brought back, including strains of Salmonella by one of the professors, also by coincidence, at Arizona State University, Cheryl Nickerson, who sent some microorganisms to space and then brought them back. I think one of the major outcomes of that has come out is that the microorganisms became more virulent when they were brought back to Earth. So that's something to consider also, you know, what will happen to just microorganisms in general as we move into space and as we go beyond the orbit of Earth, including, you know, microbiomes that are associated with our body, and any sort of plant and environment microbiomes that will be on that ship.
Thank you.
Thanks. In Zoom, okay. Okay.
Okay, Alejandro has two questions. The first one is perchlorate present all over the planet?
That's a good question. I'll answer that so that I don't lose my train of thought. Yes, it is considered that it is ubiquitous. So there have been 2 direct measurements of perchlorate, one by the Phoenix rover and one by the Curiosity rover in the Gale crater. So now basically 2 physically distant, somewhat distant locations. And then the Viking mission back in the 1970s, they also brought us some data, which at the time we did not know that was perchlorate. But now looking back at the data from the Viking missions, we also can infer that perchlorate was detected also by the Vikings. I think the interpretation of thought is some sort of other oxidant. And so it is considered that it's ubiquitously distributed over the surface of Mars, yes.
The second question is there any difference in the plant of first germinated and then
So the question is, can you germinate the plants and then put them to germinate, to put them to grow in a regolith that has perchlorate? Yes, that has been tested and the plants were not able to grow.
Is there any research on that?
So since I'll play this card. Since I'm not the plant expert, I will say I don't know, and probably I don't even want to get into that pickle. Yeah, it's maybe a question that I will pass on to the plant expert with Andrew Palmer from this project. Thank you.
And Irene Castaño has a question too. How do you try to replant the seeds from foxtail that grew in the Bioweathering system? Because many plants of that and
No. So we have not tried this, and a limitation has been the amount of bioweather regolith that we have produced at Arizona State University. So as you saw, we initially started with experiments in which we were looking something like 60 grams of regolith per bottle. We have now since moved up to do this bioweathering process, so adding the regolith with perchlorate and microorganisms at the kilogram scale. We have some bioreactors. And because the plant people need significantly more material than we have been providing, so that has been a limitation. But now we are doing this at the kilogram scale, and we will be able to answer some of these questions when there's more material available to do plant studies. By the way, it's also the most expensive dirt I've ever purchased. And so, doing this at the kilogram scale also costs thousands of dollars so it's a precious material. Okay. Thank you very much. You've been a wonderful audience. Thanks. I appreciate the question.